thank you for being here, and for those coming from Europe and Israel, thank you for still being conscious. I am going to do, as Maggie suggested, a quick journal club of sorts. I would assume that many of you have seen many of these papers, but often I'll just highlight one little thing that I believe is interesting for us Bionano users. And all of these papers are from the last few months or so, some of them from the last few weeks. This paper is a very important one. It's from Pui Kwok's team at UCSF. We have someone from his lab here. And hello.
I'm just going to get the power cord. Go ahead.
Oh yeah, power is good.
You can keep going.
Yep. And in this study, their team looked at 154 genomes from 26 different ethnic groups using Bionano technology. And what was important here was that they were really able to rebuild the phylogenetic human tree using Bionano-called structural variants, just as well as you can with SNPs. And it's remarkable how well these trees actually match. To all of us, that makes sense. But if you're not paying a lot of attention to structural variation, that may come as a surprise. What's important about this study also is that despite it being done on IRIS, our previous generation instrument, and with single enzyme NLRS, where we know we have blind spots in the genome, still in this study, they picked up 8.5 times more insertions and even a third more deletions than the 1000 Genome Project did with high depth Illumina coverage of these samples. 8.5 times. That just means that if you don't use Bionano, you just don't see really any of the important insertions in the genome. Another study done by the Structural Variation Human Consortium looked at Bionano and PacBio and Illumina to call structural variants in a reference genome. And we recolored this figure a little bit because the original one was pretty confusing with a whole lot of different categories. If you look at this one here on the left, you see deletions, on the right insertions, starting at 500 base pairs and going up to 1 mega base pairs. They're not showing the larger ones. And when you look at deletions here, you see that a large section of the largest deletions were called by Bionano only. That's what you see in blue here. And if you look at insertions, almost this entire range has a massive number of insertions that only were picked up by Bionano, even missed by PacBio. So again here, when you're not using Bionano and you're looking at human genomes, you're missing all these large variants that are the most likely to actually affect gene expression, gene regulation, and have an effect on the phenotype of your study subjects. So we were excited to see this. And again, this was done with our older NLRS technology. If you were to do that with DLS, you see a lot of 500 base pairs feeds as well that were not picked up back then. There's no such good comparison study for nanopore data. But here's a fairly recent paper from a group in the University of Antwerp, the department where I actually did my PhD in Belgium. And they used Promethion to look at NA19240. They used high coverage, 59x nanopore coverage. And they used a variety of different base callers, and so aligners and structural variant callers. And they're showing here this F measure, which is a measure that combines sensitivity and precision of the SV detection. So think of it as basically our sensitivity and PPV calls combined. And they get that to 0.70. If they did this with Bionano, it would be in the high 90s. And so in this paper, they actually claim that thousands of variants were missed or not identified or false positively called as structural variants. Thousands. And I looked, and this is the best paper that I could find for genome-wide SV calling based on nanopore data only. Their sensitivity for inversions was just absurd. They say 15% sensitivity, PPV of just 30%. I don't quite understand why this doesn't work better, but right now, at least, that's the state of the field. So I think these three papers combined just show you again that for these large structural variants, we're really the only realistic game in town. I want to talk about FSHD for a minute. You heard about this quite a bit from Mark, and we're going to hear more about this quite a bit more this afternoon. But so this disease is important to us because it's one where we're exquisitely well positioned to genotype this compared to any other technologies. And that's because we have single molecules that span the entire hundreds of kb of that repeat array, which is involved in this disease. So logically, there were a number of papers, I think 4 this year alone, from some of our users on FSHD. There's this paper here from Kunming in China and Berry Genomics, where they looked at a pedigree where multiple family members were affected by FSHD. They looked at these here, number 28, and you see clearly here the B, that is the non-pathogenic allele, present in 17 copies. You see 1 label per each repeat. And then the pathogenic allele 4QA, present in just 4 repeats here. So that caused the disease. You can measure this repeat with several different of our labeling enzymes. The data that Mark showed was with DLE1. This enzyme here, BSSS1, just happened to label once per repeat unit. So even though technically this is not better to genotype this repeat, it makes for these really pretty pictures where you just can look at that graph and immediately see how many repeats there are. And if you compare that with the standard here, Southern blot, where you're looking at messy blobs of radioactive label after a Southern blot, this is really spectacularly precise. Then they also talked about this group here of these 2 people who had a milder phenotype of FSHD. And they're not really very precise on what they think is going on, but they found a structural variant here where part of this repeat array is duplicated. This is not a standard known mechanism for this disease, but they believe that this may explain the phenotype and the fact that it's a milder phenotype. Then another paper from China, also CHOP, was involved in this, just published in Journal of Medical Genetics. Looked at 25 patients with FSHD mapped with Bionano. Again, they used largely BSSS1 in their figures because of the, I think, pretty pictures. And here's a case of a mosaic sample where the non-pathogenic 4QB allele was present in 19 repeats, but the pathogenic 4QA was present with 23 repeats and also with 3 repeats, this one being the allele that caused the disease. And because Bionano is a single molecule imaging technology, you can simply count the number of molecules that align to each of these alleles to get a measure of the mosaicism in this case. I have one slide for you that's not from an actual published paper, but I just want to use it to introduce Joe Devaney's talk later. FSHD is an easy to measure repeat for us because we have this label in every unit, or we can measure that large area. But then there's also these expansion repeat disorders, which are a fairly common cause of genetic disease, where a trinucleotide or pentanucleotide or hexanucleotide repeat expands, often expands very, very much, and causes the disease. Fragile X is an example. ALS is an example with the C9orf expansion, Huntington's myotonic dystrophy. And here's one example where the University of Florida looked at a repeat causing myotonic dystrophy in a muscle biopsy from a patient. And you can see the normal repeat here sized by measuring the flanking labels. So we don't actually have a label per repeat unit, but we simply measure the flanking labels around that repeat. That total distance is about 18 kb here. But then you can also see the expanded repeat here, which is up to 17 kb larger. And you can see this very wide distribution. And because, again, we have a single molecule technology, you can simply measure each of these molecules individually and see that distribution. And so what happens is that somatically, this repeat keeps expanding. Once it becomes unstable with every cell division, you may get larger expansions. And so some of these diseases have a late onset simply because of that. That mutation is there, but during your lifetime, that repeat becomes larger and less stable. And again, this is something that is almost impossible to measure with any other molecular method, simply because these repeats become so long that you can't get through it with long-read sequencing, especially not make sense of it with short-read sequencing. I want to talk about this paper from Jim Broach. It's in bioRxives right now, but it's being submitted for peer review, or has been submitted. Jim Broach looks at leukemia genomes, 12 genomes to be specific, and compared cytogenetic calls with this method that he developed that used Bionano and whole genome sequence. And he showed some really interesting results in this paper, including this case of a non-Hodgkin's lymphoma. They were able to culture the T cells that are unaffected and use them as germline controls, and then use the lymphoma cells, of course, as the tumor. And in this region here, on chromosome 10, you see this 3 megabase somatic inversion that cuts right through the P10 gene that's not present in the germline control. P10 is a known tumor suppressor, and there's dozens of clinical trials going on for compounds that rescue P10 mutations. This has a very clear therapeutic value for the patient, and this is something that was not picked up with cytogenetics. It was not seen with whole genome sequence. Bionano, of course, saw this immediately. Again, an example of the need for a whole genome comprehensive calling of structural variants if you really want to get the best data to decide on your treatment or prognostic options. In this paper, they show a large number of genes that have recurring structural variants in these 12 leukemia genomes. And those are leukemia driver genes. That makes a lot of sense. But interestingly, there's also a number of general cancer-associated genes that have recurring structural variants in these multiple samples. Those are not genes that are associated with leukemia. Those are not genes that traditionally are tested for in leukemia patients. And yet, there may be a lot of known, there may be a lot of data existing already about the value of these genes and these mutations, again, for therapeutic or prognostic choices. Now, on top of these cancer genes, there's also a whole lot of other genes that are not known to be associated with cancer that showed recurring variants in these leukemia genomes. Those are prime candidates for new biomarker discovery, new drug target discovery. It's just amazing to me what an enormous number of new findings there are from this single paper on just 12 leukemia genomes. I would love to see what people like you, Alex, will do with this massive data set that you're collecting on 100 or so leukemias. And then one more interesting part I want to highlight from this paper, that is that there's a significant enrichment of known cancer-associated genes near Bionano SVs, within 5 megabase pairs or so. And they're showing, this is all too small for you, but have a look at this paper. They're all showing a number of genes that are near translocations, near inversions, near deletions, much more than you would expect on average if there was no association between that structural variant and that cancer gene. So there's a lot to be discovered here, I think, about regulatory elements that control these cancer genes. There's, of course, this paper. If you'd rather watch a video, you can watch Jim Broach discuss this on a webinar that you can find on our website. We also transcribed that webinar in a white paper that's available from Genome Weapon from us. Back from cancer to human genome assembly. We just found this paper on the Tibetan genome, ZF1 they call it. And it's a spectacular genome assembly. They used PacBio, Bionano, 10x Genomics, Illumina, and Hi-C to put the genome together with a scaffold M50 that approaches the ASHG38 reference, and is even better than the Korean and the Chinese reference genomes that were fairly recent as well. I just want to point out, somewhat gleefully, that there's Bionano, 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 and OM, which stands for optical mapping, which I would assume is your Bionano work? Nope. It's fair to say that these reference genomes use Bionano for assembly for error correction. And in this particular case, they sure did. And one really interesting finding from that paper is this 660 base pair insertion that codes for improved lung function. If you decide to live in the Himalayas, then having a good lung function is pretty essential. I myself already can't sleep in Denver or Salt Lake City because of the altitude, so I surely do not have this insertion. But what's interesting here is that they found this sequence in these archaic hominids, Neanderthalers and Denisovans, and they're not present in pretty much all the other ethnic groups. So this is really a Tibetan specific for as far as they can tell. event. And sure enough, they also found more overall of these archaic genome sequences in this genome than in the other genomes that it was compared with. So a pretty exciting paper. Also exciting, 22q11. It's one of these regions that was already discussed a few times earlier today with a massive amount of segmental duplications. And these segmental duplications in this region can be as long as 1.2 megabases. There's quite a few more papers coming about this region with more Bionano data. But here they're using, again, Puig-Kwok's data set of 154 genomes to look at the ethnic distribution of different combinations of these segmental duplication haplotypes. And they see some clear ethnic differences that are quite interesting. Another part that I love about this paper is that the last 5 authors of this paper all have a sapphire. So this is quite the Bionano dream team. Ming Chao has had one for a long time. Puig-Kwok has 2. Beverly Emanuel is at CHOP and uses the instrument there. And Tamim and yours are just getting them. We got these POs in 2 weeks ago or something like that, right, Warren? Yeah. So this is quite an incredible consortium there. They're all generating data or starting to generate data, so I'm expecting a lot more on this in the months and years to come. Ming Chao at Drexel has looked at telomeres using our technology and actually developed a custom labeling method using a nicking version of Cas9 and then using our older NLRS labeling technology combined with that to label specific sequences. In this paper, he did a little bit of that, but also used, again, Puig-Kwok's 154 genome data set to look at human telomeres. And he looked at ethnic distributions of different types of telomeric and subtelomeric repeats. But he also made a beautiful description of really how our human genome evolved and which chromosomes copied which telomeres from each other. And since there's quite a few different chromosomes and they all have a couple different ends, 2 to be precise, there's a whole lot of information there about these telomeres. They did use some custom labeling, so in this case here they labeled the actual telomeric repeat to find out exactly where the chromosome ended, and they found that the references often had some extra spacing that they just couldn't find to be there in reality. And speaking of telomeres, a team led by Adam Philippi, Tina, I think you were involved in this as well, assembled a complete human chromosome. It's remarkable to think about that this is actually big news, that a chromosome was assembled from end to end. But this took an enormous amount of work. They did it in CHM13, so one of these basically artificial artificial homozygous genomes. And they put a whole lot of work in, including an absurd amount of extremely long nanopore sequencing. And I say absurd because from what I understand, that instrument ran for 9 months or so full time to get these long reads with which they could assemble that to get to one chromosome. So we have some ways to go. But I picked out this slide here where they're showing the original assembly had a contraction here of a repeat that was flagged by Bionano. You see here the Bionano map aligning beautifully on both sides, but this area was really contracted. They were able to find these long reads that could span that area and put a much better CHM13 assembly together that was perfectly supported by Bionano. You get a close-up area here which shows this 19 copy of a 9.5 kb repeat. Those are the recent papers that I wanted to show you. Again, this was just a very, very quick run through them. Read all of them if you like, and they are all on our website. If you don't want to search too hard, just go to resources, publications, and they're all listed there. You click right to the original paper. So I want to finish by talking about some of these validation studies that are ongoing. There's a couple of groups, cytogenetics groups, that really believe that Bionano could replace a number of the traditional methods used in the clinic, such as microarray, karyotype, and FISH. And we like that idea and have been working with many of them in setting up how to compare these studies. So these have all been presented at recent meetings. We have recordings of all these presentations in really high quality on our website. So to find those, go to resources, videos, and they're all there. The first one that I'll mention is from a team led by Bryn Levy at Columbia University. It's a consortium of North American cytogenetic groups from several of the largest universities like MD Anderson, Mayo Clinic, etc. And he presented results on a preliminary data set on, I think, 11 genomes that were run through the cytogenetics score at the University of Washington and then run by Bryn Levy and his team, analyzed at Columbia and with Bionano. And then the results were blind. The study was done blind, and then the results were compared. Sorry about that. And so he showed a number of examples, like this large event on chromosome 16 that was called by Bionano, but then also picked up by FISH. He compared array calls. Here was this 217 kb deletion call done on array that was a 280 kb, 18 kb deletion call by Bionano. So just incredible concordance there. And overall, he showed that there was a perfect concordance between Bionano calls and traditional cytogenetic calls. With one example, this sample where absolutely nothing matched. And based on the other results that they found, they believe that that's a sample mix-up. They're looking into that right now. Because samples were shipped from one place to the other, that happens. Here in Houston at the MD Anderson Cancer Center, Rashmi Kanagal-Shamana wanted to look at MDS. Myelodysplastic syndrome is a precursor of AML. And she looked at 7 samples where, again, there was perfect concordance between the Bionano call and the traditional cytogenetic calls. On top of that, she also showed a number of variants where Bionano really added a lot of additional information. One case here is where cytogenetics called this translocation between chromosome 1 and 12. But with Bionano, you actually see that this is quite a bit more complex. And when you zoom into that locus, you see that there's actually a part of chromosome 5 that takes part in this translocation event, something that was missed by traditional cytogenetics. And then for the last one that I wanted to show you, we just happened to have the man himself here in the house. So Alex, if you want to come up front here and talk about this for a second.
I don't know which slides you got.
I got very few slides.
Okay.
You don't need slides. I basically just want you a little bit to talk about
Sure.
this study, and maybe I need to give you this microphone too.
Sure. All right. Yeah, I can just hold it. That's fine. All right. Yeah, we didn't really practice that, so I'll try to explain you what we've done so far. So I think we got the Sapphire installed in February, if I'm not mistaken, end of February, during my AGBT stint there. We just saw the first data a little bit earlier. We sent 4 samples over, I think, last year, got the data in November, and then bought the instrument for Christmas. And I mean, I'm personally a researcher working a lot on rare disease. So next to what you see on the slide and what I'll talk about, I'm also very excited about solving some of the unsolved rare disease cases. I mean, that's really where, as a scientist, I really get excited to find things that other technologies missed, and we have the first nice examples of that. But when I was discussing this technology and the opportunities for that in-house, then also our diagnostic people directly said, oh, that could potentially be very interesting and important. So we set out to do 2 proof of concept studies in the clinical setting. One is for leukemia, where we are currently processing 100 samples. And I think we should be at 30 or so of those right now, or 35, I think. And additional 50 samples with known constitutional cytogenetic aberrations. And basically, the results we have so far is actually very much echoing what you've just shown. So far, we found all the clinically relevant reported variants that have been found by karyotyping FISH and or array. So usually for a lot of leukemia samples, we do the entire battery of tests, several FISH, a microarray, and a karyotype. And all the aberrations that were seen in more than 10% variant allele frequency, we found back. And also echoing what Sven has shown you from the other studies, quite often we indeed see, once we get closer to the truth, then we really see that some of these events are actually more complex than we initially anticipated. So even arrays and karyotyping in addition would not really dissolve the real nature of some of these aberrations. And that's what we now see in Bionano, that we believe that is much closer to reality. Some of our diagnostic people don't really like that so much because it's reality sometimes is more complex than you ideally want to write up in your report. But that is reality, and we have to find ways of doing that. And we're also using this study of the first 100 samples to find the best clinical filtering steps. So as mentioned before and shown by Alex, we can just add some of the diagnostic gene lists that we have that our diagnostic unit uses and just do additional filters on that, and then actually the complexity of the problem gets absolutely manageable. So we're quite thrilled about that. Maybe it's good to mention that in the Netherlands we're bit different from some of the labs, how they're run here in the US. So we can move from research very quickly into diagnostics. So we're doing a lot of diagnostic assays that are strictly speaking research use only, because there's a very special system in the Netherlands where diagnostics is accredited in our own system. We are ISO accredited, but it's all basically lab developed tests that we're using. So therefore we are free to use research tools in our diagnostic setup. And my personal goal is to push this into real clinical use for cytogenetics within a year or maximum 2 years from now on. Whether we match that, we'll see, but I'm carefully optimistic. And the same holds true for some of these constitutional aberrations. So I think you've seen some data earlier that some of these classical microdeletions duplications, which usually are found by CNV microarrays, either CGH arrays or CNV SNP microarrays, all of those are found back. But to also disclose all the details, we had one example where only the map found the event and not the CNV tool that's in there. That is simply that this was one of the very small microdeletion syndromes, lower than 500 kb. And we also had 2 events in these recurrent microdeletion regions, like 22q11.2, the one that Sven just shown. And there we find the event by the CNV tool, but the map didn't initially support it. Now we worked with Alex and his team, and Alex just sent me beautiful slides by just using, I don't know how much percent, but only the reads that have a certain size, so using the longest molecules from your assembly, then you can assemble even across those regions, which is quite remarkable, as you've seen the complexity also from Joris Vermeer's paper. I mean, that was the most important bits that I had in to tell you probably.
What about this line on top? We have this printed on our booth, by the way.
Right. I mean, that truly is the case. I'm originally trained in a cytogenetics lab, and I always admired the people that were really good at karyotyping. It really is much more of an art than an accurate science. There's some people that are so skilled at looking at chromosomes, but it requires years of training and a certain amount of talent that not everybody has. So it was never a really objective way of doing it, I think. And I have to say my old cytogenetics love really got revived this year by having Bionano in-house. I think there's still a lot of things, and Claudia and the other presentations this morning shown, even for the well-understood loci of the genome, we don't fully understand them yet because there was no technology to decipher those. So I think that is really where some of my enthusiasm comes from, that we now for the first time can really look at individual DNA molecules that are really large, and that is quite exciting. What is not on this slide, I think we have the first research findings that are really cool. So we collaborate with former PIs of our department, and I think it's summarized in one of the videos. So for example, we had one case with a teratoid tumor, so very severe childhood cancer, and unfortunately that child already died of it, aged 2 years. But that family wanted to know whether there is a recurrence risk for that event. So that is a classical diagnostic request, and you have a suspicion for a germline mutation in a gene called SMARCB1. And that family, or the DNA of that child, was sequenced by Sanger sequencing. It was studied by MLPA. It was done by exome sequencing, all in a diagnostic setting. And unfortunately, the mutation was never found. And in addition, that group also did genome sequencing, standard short-read Illumina sequencing. They didn't find any mutation. So it remained very unclear whether there's a recurrence risk or not. And now we did Bionano optical mapping, and we found a small 2.4 kb insertion that was really not found in any of the other technologies. And honestly, we can't even validate the event yet, because even just PCRing over that locus is not trivial. We always just get one amplified molecule so far. So I think there is some funky events. I personally think it's an insertion of whatever, 99% GC or so. And those remain undetected with any technology because everything is based on PCR and sequencing and so on. Whether that is resolving the the remaining, whatever, 1% or 10% or all of our unsolved cases, I don't dare to say yet. But we have quite some exciting new cases that really support that this is really adding to our sequencing portfolio. Was that okay? Is that good?
That's great.
All right.
You're hired.
All right. I have a good job. I don't need to be hired. I'm happy.
I mean, as our spokesperson. So that's all I had for you today. If I've missed any papers that you think we should present, send them our way. And thank you for your time, and don't try this at home.